as you know. Let me turn this on. My name is I think it was on. My name is Carlos Roque. I'm from the USP-Ribeirão Preto. I work on computational neuroscience. I'm going to talk these 3 lectures, one today, 2 tomorrow, on this subject, which I believe for most of you should be new. So I prepared this talk to be a kind of broad overview of this field. I won't have time to go into that much detail in my talk, so I'll give this overview and show you how to model neurons, synapses and networks, and hopefully try to present something of my own work at the end tomorrow. So I'd like to start by thanking the organizers for inviting me to give this course here. This is not my first time here at ICTP. This is a great place to do science, to do theoretical physics. So it's really a pleasure to be here and share this talk with you. So to start with, since I said I'm in Ribeirão Preto, this city here in the state of São Paulo in Brazil. There we have the University of São Paulo. This is the campus. Here you see the Clinic Hospital, which is probably the best, well, one of the best research-oriented medical schools in Brazil. I am in the physics department, which is somewhere hidden under the trees there. And this is the city of Ribeirão Preto, and this is the Department of Physics at the School of Philosophy, Sciences, and Letters there. And my group, as it was, say, 2 years ago, now some of the students have left, some came, but I don't have any new picture to show you. Anyway, it's about this size, still, the group there. Okay, so I divided this course in several parts. The first one, part 0, is just a presentation of computational neuroscience to give you a gentle introduction to the subject. So for most of you who are physicists, I believe all of you are physicists, I'm a physicist as well, neuroscience is a very new subject compared to physics. When we start studying physics, we hear about, say, Galileo in the 16th century, Newton, 17th century. When we moved to neuroscience, the oldest, of course, there were people talking about the brain before, but when the science itself started was at the end of the 19th century. So it's quite new compared to physics itself. The concept of neuron was proposed in 1889 by Santiago Ramón y Cajal, a Spanish neuroanatomist. The very concept of cell was really very recent at the time, and he used Golgi staining techniques, a technique that was proposed by Camillo Golgi from Italy, to stain neural tissue, and then he observed that the neural tissue is made of small cells, as other cells on other parts of the body. And he wasn't the one who coined the word neuron, but he developed the concept. A few years, still at the same time, or 10 years later, Sherrington in England coined or proposed the concept of synapse, which is the contact point between two cells, two neuron cells. But the very word neuroscience is very new. It was the first official use of it is from 1965, so it's even younger than myself. Before that, people had neuroanatomy, neurophysiology, neurobehavior, but not really a field, a science itself that would encompass all of those approaches to understand the nervous system. The first Society for Neuroscience, which is the American Neuroscience Society, the biggest one in the world, was held in 1971, and it had about 1,100 participants. The 51 last year in Chicago had over 30,000. I think it's the biggest conference I've ever been, larger even than the March meeting, the physics conference. So it's a quite, say, new and a fast expanding subject. Well, the brain is probably is the complex system in the universe, if you want to consider that. Is probably the ultimate example of a complex system, not only because of its sheer size, the number of neurons and synapses, but by the complexity of the connections between the cells and the complexity of interactions spanning different orders of spatial and time magnitude. So as I said, here is only for space, but you could have a similar diagram for time as well. You have phenomena related to brain and neuroscience at the molecular level. all the way to the whole body, whole system level. All of them interact in a way or another to create what us are. No? That's why the brain is so important, because basically the brain is us. Since the brain is very important, there are lots of, experimental investigations on it using different techniques. So there are lots of data on the brain. Anatomical data, physiological data, biochemical data, psychological data, and more recently, molecular biology data, imaging data, optogenetics data, and mathematical and computational modeling data. So theoretical physicists and theoretical scientists in general enter the field more or less, say, in the past 50, 60 years, and started to develop what today is called computational neuroscience, which is considered among the neuroscientists as a research technique to understand the brain. So today people can say that people who do computational science do in silico modeling to compare to in vitro and in vivo approaches to the brain and biology itself. So you basically simulate pieces of neurotissue in computer in silico, and then compare or make predictions about the behavior of the real thing, the biological stuff, the wet stuff. So what is computational neuroscience? Well, computational science is the theoretical part of neuroscience. It's also called theoretical neuroscience for obvious reasons. Its objective is to determine and articulate the principles of mechanisms behind the functioning of the nervous system, as any theoretical science does to its subject. But the novelty here is that it is assumed that this functioning involves some sort of computation. I won't define computation here, I don't have time for that, but you can imagine that computation in this sense has nothing to do with the computation that the computers that we use do, but you can imagine that computation has a more generic definition. I'd like to define computation as a structured manipulation of information in a state-dependent way, and also in a transition-dependent way, in the way that when you move from one point to another one, the computation changes. So in this broad, say, acception of the word, the brain does computations, and that's why the name of the theoretical part of neuroscience was priced as computational neuroscience, to emphasize this, that we want to understand the computations that the brain does. Of course, due to the complexity of the brain, most of the models that we construct have to be simulated in computers. So many people also think that computational neuroscience means that we use computers to simulate the brain. Of course, this is one of the reasons why the computation word is there, but the real reason is that we want to understand the computations that the brain does. Okay, now after this very brief introduction, let's go to the basic concepts, because since you're physicists, you need to be introduced to some of the concepts that involve neuroscience. Sorry?
Just to let you know, there are physicists, but there are also chemists, there are some engineers.
Great, thank you. Yeah, so for some of you, this is really old stuff. But for the physicists and for the engineers, this is new stuff. Okay, so you can ask the biologists around to have, say, further information on this. Thank you. So then the neuron, the cell that was proposed by Cajal, Santiago Ramon y Cajal, is a cell. They are isolated as any cells in the body, and they are not the only type of cell in the brain. There are the glial cells, which occur in larger number than neurons. By far outnumber the number of neurons in the brain. Their function traditionally was considered to be just of, say, nourishing support, but nowadays, more recently, people are starting to perceive, to understand that glial cells also have functional roles in the brain, and they are studying glial cells. So it might be that in 100 years' time, people will give a different talk about neuroscience, and they will emphasize glia instead of neuron. But at the moment, neuron is the big thing. The cell neuron is the real thing. Okay, so we're going to focus on neurons in this course. They are, as any other cell, they're structurally, metabolically, and functionally independent. according to the neuron doctrine, which was postulated by Cajal over 100 years ago, the neuron is the basic functional unit of the nervous system, different from the glia. And the interesting thing is that they are specialized for neurocommunication. That's why we talk about computations in the brain. So the structure of the neuron, this is the generic, say, textbook-like depiction of a neuron, is that it's a cell. It has the following parts. It has dendrites, which is the input part of the brain, where a cell receives input from the other cells. There's small processes close to the cell body. And there's the soma, or the cell body, where the nucleus is and all the organelles are, which integrates inputs. And then there's a large process emanating from the cell body, which is the axon, from which output goes to other cells. And this output reaches other cells via synapses, which contact specialized structures that connect, say, two cells in the brain, and they're called synapse. That was just the textbook example, but if you study neurons, the real neurons, you see that they come in different shapes and forms. Here, 3 of them. But in general, all of them have dendrites. Some of them have profuse dendritic arborization, as the Purkinje cell. Some of them have more simple ones, but all of them have dendrites, the cell body, and an axon. Oh, not all of them. There are neurons that are axonless, and very few neurons are also, they don't have dendrites. But the generic structure works, especially for the central nervous system. Here more structure, so that you can see that the shapes of the neurons are really varied, and we still don't know the functional role of these wonderful, the beautiful shapes that neurons can take. Also, neurons are connected into circuits. I will use the word circuit for like a local network, or like a regional structure in the brain, and network for the brain-wide network, for the whole brain network. So they're locally connected and globally connected as well. As any cell, the neuron, the cytoplasm, the interior of the neuron is separated from the extracellular space by a membrane, the neuronal membrane, which is a thin membrane composed by lipids and proteins immersed on these lipids that cross the membrane from one side to the other one, and which act as channels from which, or through which, ions can flow, both towards inside or outside of the cell. They form channels, as you can see there. So those ion channels, they can close and open depending on the external voltage, the membrane voltage, the local voltage at the region where the cell, where the ion channel is, and also due to binding to some substances in the extracellular or intracellular mediums. Here's a scheme showing just the protein ligand binding, which leads to opening of this channel. One of the very basic facts about the neuronal membrane is that there's a difference of electric potential between the inside and the outside of the cell. So if you ground the extracellular space, you say that the extracellular space is at 0 potential, and if you insert a microelectrode inside a cell, you'd notice if you did this experiment that there's a difference of potential between the inside and the outside. And the difference of potential is negative, first fact, and is of the order of magnitude of, say, minus 60, minus 70 millivolts. So there's a difference of potential of about minus 70 millivolts between the inside and the outside of the cell. This is the membrane potential. Also, the membrane separates ions from the inside to the outside, and they come in different concentrations. The concentration of a given ion, say sodium, is larger in the outside than the inside, and of potassium is the opposite. It's larger, more potassium inside the cell than outside the cell. And chloride as well is more concentrated outside and inside. It's easy to remember because the external medium is a saline solution, so NaCl dissolved inside the extracellular spaces. Here, some numbers, some figures for the frog mussel, the squid giant axon, and the typical mammalian cell at their respective temperatures. So the membrane it separates ions from the two sides, and the concentrations are different. This is the main fact here. So what's the origin of the membrane potential? It's basically due to diffusion, basic diffusion, that since there's a difference of concentration and there are channels specific for different types of ions crossing the membrane, say potassium in this case, of a given ion, flows against the concentration gradient to the other side. But since the ions have charge, this creates an excess of, say, in the case of potassium, positive charge to this side here, which is the outside of the cell, an excess of positive charge here, and deficit of positive charge there, an excess of negative charge, which creates an electric field, which drives potassium ions to the opposite direction. So basically, there's a competition between two driving forces here, the diffusion and the electrostatic driving force. Initially, the diffusion wins, but as more and more potassium flow to the other side, potassium ions flow to the other side, the electrostatic drive forces, the dragging force, electrostatic driving force forces potassium to the other side, and eventually the two of them enter into equilibrium. They equal itself. And at this point, there's a voltage that defines this equilibrium, which is so-called Nernst potential, or Nernst voltage, because it was calculated by Walter Nernst, the German physicist and physical chemist from the 19th century, and this is the equation for it. So it depends on the gas constant, the absolute temperature, and the charge of a mole of ions, and also, and most importantly, on the ratio between the concentration of the ion from the outside to the inside. So here are values of the Nernst potential for different ions and the standard conditions for the squid giant axon, which was the model system used by the first neuroscientists, first biophysicists who studied this problem in mid-20th century. So the equilibrium potential for potassium is minus 75 millivolts. This is if the only ion that could flow across the membrane was that ion. So if only potassium could flow across the membrane, the equilibrium voltage would be minus 75. If only sodium could flow, the equilibrium potential would be plus 55, and so on. These two are the most important ions for cells in the brain, potassium and sodium. So you ought to remember those numbers, minus 70 something for potassium and plus 55, plus 50 something for sodium. So one is negative and the other one is positive. When, in a real situation, that the different ions can cross the membrane at the same time. The membrane is not only, say, permeable to a given ion, but it's permeable to all ions. In that case, one can still calculate the equilibrium potential. That's the work of Goldman, Hodgkin, and Katz in the 20th century, more or less in the 40s of the past century. And the equation is called the Goldman-Hodgkin-Katz, or GHK equation. It's given by this expression here, and is written in terms of the permeabilities, not of the conductances of the membrane to the ions. So the ratio of the permeabilities of all the ions to potassium, because the membrane under resting condition is more permeable to potassium than to any other ion. So potassium is the reference ion there. And so if you plug in the values that are measured experimentally, you observe that the equilibrium value for the squid giant axon at 20 degrees Celsius is minus 70 millivolts, which is exactly the value measured experimentally. So you, by the way, just can remember that the resting membrane voltage for the squid giant axon is very close to the Nernst equilibrium voltage for potassium itself, which means that under rest at equilibrium, the membrane say conductance is dominated by the conductance of the membrane to potassium, not to the other ions. This is very important. Second experiment that one could do instead of just measuring the difference of potential would be to stimulate the cell at the point where one is measuring the potential, the voltage. And if you stimulate by a current, there are two things that can happen. Either this current is negative, which means that it takes the voltage to more negative values than its resting value. Sorry. Yeah.
Just a couple of slides ago, what initiates that diffusion from one side of the cell?
What initiates? Just the difference of concentration. You have, say, more potassium this side to that side. If you have a channel open, and the diffusion is a passive process, passive phenomenon. So you will diffuse. Yeah?
You don't end up with the same amount of potassium on both sides.
No, because this is a membrane phenomenon. You imagine that there's both solutions are neutral there because you have organelles that have all the structure which are charged. So the overall charge, both in the intracellular and extracellular, is zero. This is just a membrane phenomenon. A very tiny fraction of ions of charge really flows across the membrane. Okay. So you can inject a current that takes the voltage. So yeah, this is the current. I'm representing here as negative, different degrees, different step sizes of current. And then all of them, in this case here, they take the voltage to more negative values with respect to the zero there. We call this hyperpolarization because it increases the polarization of the membrane. And what happens in this case is that as you increase the amplitude of the hyperpolarizing current, you increase the amplitude of the response in a graded way. So there's nothing really new about this phenomenon. It's quite easy to model this. And you can see here that there's curved lines there. Anyone would guess what's the origin of those? Say the response is not instantaneous as the one of the injected current. There's some time involved there. Anyone would be able to guess what's the origin of this? The capacitance, yeah, it has to do with the capacitance. The membrane acts as a capacitor, so this is the typical response of a capacitor when it is charged. But a very different thing happens when you inject current that takes the voltage towards zero. So you depolarize the cell. You reduce the polarity of the cell. In this case, at the beginning, you have more or less the same or a similar phenomenon. So you inject the current, and you have a graded response. But whenever you have the current reaching a so-called threshold point, and the corresponding voltage depolarization reaching a threshold point, which in this picture here could be more or less around here, you have a stereotypical phenomenon that cannot be controlled by the experimenter. The voltage starts to grow very quickly, and in about half a millisecond, it goes to a zero and crosses zero and becomes positive instead of negative. So the inside of the cell becomes more positive than the outside, and it goes to very high values there, say plus 40, plus 50 millivolts. It stays there for very short time, less than a millisecond, and then it decays very sharply to a value that is negative again and overshoots the resting value by some amount there, and then slowly recovers. And if you do this in an axon, like the one I'm drawing here, this so-called action potential propagates to both sides. If you inject a current in the middle of an axon, you can generate a pulse, a voltage pulse that propagates to both sides. This is called action potential because it acts in anything that's at the ends of those axons, especially if one of them is at a synapse. So that's the name, action potential, also called spike. Okay. So if you depolarize the cell, you have this very different phenomenon from the other one. And this is the origin of all the wonders that you have in the brain and in ourselves, the fact that the brains can communicate via action potentials. So that's an action potential, a spike. For each specific neuron, it has a specific shape, so it's like a fingerprint. For each neuron, have a specific shape, and the amplitude and rising time, decay time, hyperpolarization peak. All the features are specific features of each cell. So if you have a stimulus here, you have the depolarization phase, and then you see here time is in milliseconds, so it's very quick, and then a peak, and then a repolarization, the hyperpolarization phase, which is also which corresponds to the so-called refractory period, which is a period in which it's very difficult to stimulate a second action potential, and then the recover to resting state. If you stimulate a cell with subthreshold depolarizing currents, you cannot generate action potentials. You just generate potentials, pulses of potentials that propagate, but they dissipate very quickly, and they cannot propagate over large distances. On the other hand, an action potential can propagate over long distances, say from my brain to the point of my toe and back, say distances of an order of 1.5 meters and so on, without dissipation, without changing the shape at a velocity which is about 25 meters per second. But it depends on the type of cell. Okay. So as I said, there's a refractory period. What we can imagine that as a period of time in which that threshold, that's the threshold for the initiation of an action potential, is infinite. So since the threshold is infinite, an action potential cannot be generated during the so-called absolute refractory period. And then there's a relative refractory period, which is a time during which is, again, possible to generate an action potential, but you need a stronger current. And the strength of the current, the amplitude of the current decays with time until after the end of this time, the initial current that you used to obtain the first action potential now can also generate an action potential. So there's a refractory time there in which initially it's impossible to generate an action potential, and then it's more difficult but still feasible to generate an action potential. Well, as I said, there's a chemical transmission across synapse. There's a synapse that connects the two neurons. In the case of a chemical synapse, you have this cartoon here showing all the elements of a chemical synapse. So the spike, the action potential, propagates down the axon, and it reaches a synaptic button where there are vesicles containing substances called neurotransmitters. Vesicles diffuse through the intercellular medium. They hit the membrane from the inside. They fuse with the membrane, and they release neurotransmitters in the synaptic cleft, which is the space between the presynaptic cell and the postsynaptic cell. There's a second diffusion process now. The neurotransmitters diffuse across the synaptic cleft, and they bind to receptors, which are proteins in the postsynaptic cell. And the binding in those receptors make those receptors open, because the receptors are proteins like the ion channels that I've shown to you before, and the opening of those receptors allow the entrance of ions inside the postsynaptic cell, which can depolarize or hyperpolarize the cell. And these are so-called postsynaptic pulses, and they can propagate. They're not action potentials because they're much smaller than an action potential, but they can contribute to the generation of an action potential. Okay? So this is the generic scheme of a synapse, chemical synapse. So as I said, synapse generates postsynaptic potentials, which are tiny fluctuations in the voltage, in the membrane voltage of the postsynaptic cell. They can be depolarizing, in which case the synapse is called excitatory synapse, and they can be hyperpolarizing. They can make the voltage more negative than the resting value, and in this case the synapse is called inhibitory synapse. So synapses can have, say, two flavors if one. They can be excitatory or inhibitory. A given synapse is either one or the other. You cannot have a synapse that's both excitatory and inhibitory at the same time. That's because the neurotransmitter that a given cell, a given synaptic terminal releases is always the same. So synapse can be either excitatory or inhibitory. This is a principle that was observed experimentally by someone called Dale in the 20th century, and it's called Dale's principle.
But if considered two neurons, can one communicate with the
Of
other
course.
by both inhibitory and excitatory action?
A given neuron only releases the same neurotransmitter. So a given neuron is always either excitatory or inhibitory. But you can have, say, two neurons. You have a neuron there. One is excitatory, the other one is inhibitory. So one can excite this neuron, and the other one can inhibit. Yeah. Okay. If you study electrophysiologically the types of firing patterns that a neuron has, you find that they're very diverse, different types of firing patterns. So you see most of them here, not all of them, but most of them are initiated by a constant injection of current with a given amplitude, so-called degree input. And the response is different depending on the type of neuron. So some neurons have tonic spiking, so they spike after some adaptation period. They spike in a tonic way, like ta, 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 ta. Some of them have just a phasic spiking. They spike just at the beginning when the current is turned on, ta, and then they silent. Some of them have bursting behaviors like trrr, 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 and so on and so forth. So neurons don't respond always in the same way. Neurons have so-called they belong to electrophysiological classes. And these behaviors are due to the ionic channels in their membranes. The dance of the ionic channels that is what generates the different firing patterns of each neuron in the brain. This axis here is time, and this is the voltage, so millivolts. This, by the way, is a simulation of all the patterns. This is not a real recording from real neurons, but it's a simulation that captures very well the behavior of the cells. But here would be voltage, and here time. Okay? Because these are action potentials. So the voltage is here at the rest. When there's a stimulation, a current, the voltage increases, generating an action potential that repolarizes, and then there's another one that goes up and then down, and so on and so forth. So this could be, say, hundreds of milliseconds. So you see the action potential mostly as a vertical line, but each action potential there would be something like this. It's a rising phase and downwards phase. This is an action potential at the resolution of, say, a few milliseconds. In these plots here, this axis here would be measuring time in, say, hundreds of milliseconds. So in this resolution, you basically would see vertical lines. But all of them would be action potentials. Since at this resolution you see basically action potentials as vertical lines, one can imagine that the brain or the neurons use those action potentials to communicate. And since all of them are stereotypical, what matters there is say the frequency of them or the actual timing of those spikes during a given time. So you can have different so-called neurocoding schemes, which are ways in which you can arrange those spikes to communicate or to convey information to other cells. So you can have, say, so-called rate code based on the rate. So you define a time window, a short time window, and you basically count how many spikes occur during that time window. If you, in the same time window, change the rate changes, you can say that the neurons are coding things by the rate of discharges of spikes. Also, you can imagine that the actual time of each spike is what matters. So the timing itself, if a neuron receives this or emits a spike now or 1 millisecond later or 2 milliseconds later. So the time itself is important. And also, if you have, say, more than one cell present, that you can have a kind of correlation code in which the correlation between the spikes is what matters. Say, the synchrony between them. These are just ideas. Of course, we don't know how the brain communicates. Even the idea of neuron coding may be a wrong idea, because the idea of code is something that remembers us of, say, two people that arrange a code to communicate, and the brain doesn't have that. But it's a, say, fruitful idea for us to analyze, offline at least, the behavior of spike trains, idea of neurocoding. Here are some examples of rate code, because then after people had those ideas, they started looking at those types of coding in the brain itself. And they found that the 3 types of coding exist actually in the brain. So here are some examples of rate coding. So this is coding of skin pressure. So you have skin pressure receptors in our skin. They're called Merkel corpuscles, not due to the Angela Merkel, but they're called Merkel due to some Merkel who discovered them. And if you just press those receptors, which are all over the skin, and measure the response of the fiber that come from those cells, and count the frequency using a short time window, you see that they can fit more or less well by a straight line. So that actually the rate, the frequency of firing codes the intensity of the stimulus for this case. You can also, I don't know whether you know, but in the visual cortex of mammals, cats and primates as us, we have cells that are sensitive to orientations of light bars projected on a screen. So if you project on a screen a light bar with a given orientation, and you measure the response from cells in the visual cortex, here at the back of the head, you see that some cells, as you have here, are sensitive to the orientation. So the particular cell shown here is sensitive to this vertical orientation there. So the rate of spike is larger for the vertical than to any other orientation. So that if you plot a curve like that, you have a kind of tuning curve for the orientation to which that cell is sensitive. Here you have another example. There are cells in the cerebellum that are sensitive to the angle, to the joint angle, to the movement of the joint angle done by a subject. Depending on the joint, you see here, the movement occurs during this time window here. And depending on the type of movement, the rate, the frequency of firing is higher or lower. So there are some examples of rate code in the brain. There are some examples of temporal coding, say, for example, bats using the precise timing of spikes to echolocate to create a representation of the space surrounding them. That's how they avoid us. For example, if you go, say, I don't know about here, but the University of São Paulo campus, in the evening, you'll see lots of bats flying, and you can walk around the trees there without even noticing that they are flying, because they can avoid you, because they are quite good in this. Us as well, we have this binaural sound localization. There are some delay lines in our brain that create delays between inputs from the left and right ear, and so that when 2 inputs come, say, to a given cell with no time delay, the brain assumes that they are at equal distances. So you can use this to locate objects in space. And there's also examples of correlation code. This is more an assumption than really a measurement, but there are other instances, but they'll be very difficult to explain to you. So I use this example to convey you the idea. That's the binding problem. Say, when you watch a complex image like this, a woman and a dog, you see the woman is wearing a green shirt, blue pants, the dog is white. How can you say that the green is the color of the shirt that the woman is wearing, the white is the color of the dog, and not vice versa? How can you bind? Because when we look at a picture, our brain, the initial stages of processing, it separates everything. There are neurons that process color, neurons that process, say, movement, and so on, texture, and so on. But at some point, at some higher order processing stage in the brain, you bind all of them together to give the impression of a woman or a dog. How do you bind these? One of the ideas is that the binding is due to synchrony. So groups of cells that are firing synchronously at a given time represent the woman, and another group of cells represent the dog. And there are cells that represent the color as well, and so on and so forth. So this would be an example of synchrony-based code. Well, so now just to give you some examples of possible mechanisms that one could have just by connecting cells in excitatory and inhibitory ways. So one of the mechanisms is so-called lateral inhibition. It's a mechanism in which cells inhibit whatever is to their sides. So a cell inhibit cells to their right or left side. And this could come in two different ways, so-called feed-forward, in which the inputs themselves inhibit the cell. So there's an input here that would excite the cell, and the input would inhibit the neighboring cell. And the same thing with this input here. The input here would excite the cell and inhibit the other one. So this would generate some form of inhibition, of lateral inhibition between the two cells. But also one could have feedback inhibition, in which inputs excite both cells, and the cells themselves, they have axons that would inhibit the neighboring cell. So this cell would inhibit the neighboring cell, and these also would inhibit the neighboring cells. So one could have two different ways of having lateral inhibition. And they could be implemented in real nervous systems. So in the 50s, these guys here, Ratliff and Hartline, they were studying the horseshoe crab, very tiny kind of crab. I think it's common in North America. It's called Limulus. And its visual system is composed of ommatidia, which are cells that are separate from each other, and they receive light, and they convey the impulses to the central nervous system of the animal. And by studying, and they found that the system inhibit, one ommatidium inhibits the neighboring ommatidiums, and they wanted to know what type of mechanism was that, feed-forward or feedback. And they using some clever experiments, they were able to see that the mechanism is feedback. I don't have time here to describe all of this, otherwise I'll spend a long time. But you have access to the PDF file, so you can read all the details here and see that it really it's feedback. And then what's the functional role of this lateral inhibition in the visual system, for example? It can generate contrast enhancement. So it enhances contrast between, say, one part of the visual field of view and another part. And there are even optical illusions due to this phenomenon that you can search in the internet. Also, if you couple cells, excitatory cells to inhibitory cells, one thing that's very easy to obtain is oscillations. Here's an example. Suppose an excitatory cell receives excitatory input from a pattern generation that generates a regular, that adds regular spikes, inputs to them. So this cell gets excited, so it fires, and since it excites this inhibitory cell here, it prompts the inhibitory cell to fire as well. And then the inhibitory fire feeds back to the excitatory cell. But when the inhibitory cell does that, it inhibits the excitatory cell. So it shuts down the excitatory cell for some time. Since the excitatory cell is shut down, it stops exciting the inhibitory cell, and then the inhibitory cell stops inhibiting the excitatory cell, and then the excitatory cell, which is receiving constant input from the other cell there, starts firing again, and so on and so forth. This mechanism repeats. So it's a mechanism, very simple mechanism that generates oscillations, and oscillations are ubiquitous in the nervous system. You can find both in circuits and brain-wide as well, like the EEG measurements that I showed there. Another mechanism that's very important is synaptic plasticity. Synaptic plasticity is just a generic name given to any type of change in the synaptic strength between two cells. You can imagine that the synaptic contact has some, say, sort of efficacy, some sort of efficacy measure of how efficient, how efficient is this, say, the presynaptic cell is in exciting, in conveying the information to the postsynaptic cell. This is generically called synaptic strength. And the synaptic strength can change over time, can become stronger or weaker due to several mechanisms. So the generic name given to this is synaptic plasticity. And they can last for short or long durations as well. And this is the hypothetical mechanism underlying memory and learning. So it's very important for us to survive as a species, to learn things. in the 1940s, the psychologist here, Donald, had proposed a mechanism that would explain synaptic plasticity, which today is called hebbian plasticity. So imagine that you have two cells connected by a synapse, which initially is very weak. The strength is very weak. But suppose that the cells fire at the same, more or less at the same time. So they fire together. Say the presynaptic cell fires and the postsynaptic cell responds. Due to this, say, kind of synchrony mechanism, the synapse is a little bit increased. It's like when I'm talking here. The student who is paying attention to me is more or less like in synchrony with I'm talking. So we create a strong bond between us. The student who is not paying attention doesn't care, so we don't create a strong bond. So there's an increase and weakening between our connections. It's more or less the same thing. The presynaptic cell spikes and the postsynaptic cell responds, so there's an increase in the synapse. And then again, the presynaptic cell fires and the postsynaptic responds. Notice that this implies action in both cells. The presynaptic has to fire and the postsynaptic also has to fire. Like when I say something and the student has to pay attention to what I'm talking. So it requires both. It's a joint mechanism, a synchrony mechanism. And then the synapse gets even stronger and so on and so forth. The two fire and the strength of synapse grows with time. So people call this, in English they have this expression here, neurons that fire together, wire together. I think it conveys very well what is meant by the hebbian plasticity rule. So neurons that fire at the same time, or more or less at the same time, in a correlated way, they wire together. They create strong bonds between themselves. So this was proposed in the 40s by Donald Hebb before people even knew how cells generate action potential. So it was just an idea. Later on, it was found people have done experiments with real cells, and they found that there are some candidates, some physiological candidates to hebbian plasticity that can incorporate, can instantiate the hebbian plasticity mechanism. So it's very important as a conceptual mechanism for us to imagine what can happen in the brain. So just to give you an example of what we can do functionally with this, what would be the function of hebbian plasticity? So I'll show you using a very simple example. The example that I give in popular science talks that I give, say, to secondary students, how one can improve, how can one implement associative learning. I think you all know what associative learning is. I learned to associate one thing to another one. It's a very basic and very important type of association that we use all the time. If you want to remember something, oh, I remember, what's the title of that movie I saw 2 weeks ago? I don't, oh, but the actor who was there was this guy. Oh, yeah, because I remember the name of the actor, I can remember the name of the film, and then I can remember the plot. So I make associations. So imagine I have a group of neurons there somewhere in the brain. And then you go to a movie, and then you see Star Wars, for example. And then your favorite character there, Chewbacca, is there at a given moment. And the image of Chewbacca activates some cells, say, in your visual system. So they are activated just by the image of Chewbacca there. And then it disappears from the scene. Another character enters in the screen. And so the neurons which were active when Chewbacca was there, their firing starts to decrease because the stimulus is no longer there. So the stimulus is decaying until zero. So this shows that the firing of the neurons itself wouldn't be a good mechanism for, say, memory storage in the brain, because they are present only when you are watching or hearing or in the presence of the stimulus. After the stimulus, you don't have this anymore, so you don't remember it. So how can you remember things for longer times? So imagine that when you were in that movie, you were, say, eating an ice cream, a blue ice cream for that matter, and the blue ice cream has a very diffuse projection across the nervous system of the person, and it so excites different types of cells all over the brain. In particular, they excite some cell that has connections to those cells in the, say, visual system area, or in, say, in the memory area of that person who was watching the movie where Chewbacca was. So imagine that at that time, the person was watching the movie and he or she saw Chewbacca there and was eating the ice cream. So those cells were activated because of the presence of Chewbacca, And this cell here was activated because it represented the ice cream, and it has connections to all of the neurons there, which are very weak because they were not, say, built to represent ice creams. But suppose you have a hebbian mechanism there happening. So if you correlate the image of Chewbacca with the eating of the ice cream for, say, some seconds, or for as many times you watch the movie and eat the same ice cream, you increase those connections between this cell here and the cells that represent Chewbacca. Only those. The other ones don't have their synaptic connections strengthened. So you imagine that you repeat this, because there might be some, say, emotion mechanisms also involved, some mechanisms that collaborate to intensify those actions. But just the hebbian mechanism is enough. Just by correlating the activities of those cells, you increase the strength of those synapses. That's why they are represented by increasing the width of those lines there. And imagine that the strength of synapse decays with a much longer time scale than the activity of the cells. So while the activity of the cells decays in milliseconds, the strength of synaptic weight decays over minutes or days or years. Okay? And then, yeah, at the end, we'll say you stopped watching the movie and you stopped eating your ice cream. So the activity of the cells that represented Chewbacca and the ice cream decays, but the synapse also decays, but at a much longer time scale. It takes much longer for them to decay. So after, say, days after you saw that movie or you ate that ice cream, the activities of the cells are no longer there, but the strengths, the synaptic strengths are still there. And the synaptic strengths represent the connection between the ice cream and Chewbacca. And imagine that, say, 1 year later, you were walking down a beach, and then you see someone selling ice creams, and then you buy the ice cream, just because you like ice cream. And then you go there, start eating the ice cream, and suddenly, wow, you remember Chewbacca, and you remember that day in which you were watching the movie, and so on. You associate something. Just because the activation of this cell activated the cells that represented Chewbacca, because the strengths are strong. No? The synaptic strengths are large. Okay? So this is a mechanism, and then you remember that. No? So this is a possible mechanism of associative learning, just because of hebbian learning. A very simple example, but I think it conveys, I think you can understand quite well what I meant by that. Okay? So what are the components of model in computational neuroscience? Because what we want to do is to model things like that, to construct models that allow us to understand how memory is processing, how emotions, how the role of emotions in the brain, and so on. We want to understand ourselves. That's the ultimate goal of computational neuroscience. So you construct a model. The model has an anatomical part which corresponds to the structure of the system itself, how you, what type of cells, how you assemble them, and also has some dynamics, which means what type of activity those cells generate. And the activity is due to the way I connect them, but also due to the intrinsic characteristics of each cell. So the dynamics is some property that emerges from the structure that you put in your model. And by structure, I mean just not only the cells themselves, but the way I connect them. And since you have plasticity as well, you must have plasticity in a good model in order to be able to emulate things that happen in the brain. And the plasticity has to do to the dynamics, the way the neurons activate. The activity of the neurons themselves, the dynamics of the system changes the way the elements of the structure are connected. As I said in my example, my previous example, it changes the strength of synaptic weights. And this is plasticity. And this changes the structure itself of the system. So you have this complex structure here in which structure influences dynamics and dynamics influences structure. So that's what we want to capture in a computational neuroscience model. To construct a model, one needs basically 3 elements. So that's why I have this 3-dimensional representation of a network model in computational neuroscience. You have to choose a neural model or some neural models, because you can have a heterogeneous type of model in which you have, say, neurons which are simplified, neurons which are more biophysically realist. Doesn't matter. I have to choose some neurons, some models of neurons to put in my model. I also need to choose models of synaptic connections. How I'm going to model the synapses in my network. So I have in shelf of synaptic models, very different types of them, and I choose some of them to put in my model. And I also need to choose an architecture for my model. What type of architecture? How I'm going to structure those neurons and the connections. Is it going to be a random architecture? Is it going to be modular architecture? Is it going to be inspired on the data from the brain? Just going to be a toy model? So I have to choose the necessary structure as well. So having those three elements, I can construct a network model. So any network model is composed of a neural model, synaptic model, and architecture model. And I need to specify them before I construct a model. And in the next two lectures, the 2 lectures of tomorrow, I'll go through those three, features there. I'll present to you types of neural models that one has today in the market, types of synaptic models that also one has there to choose, and also types of architectures. And hopefully at the end, I'll have time to show you at least one example of this type of model. If not, at least you'll have all the elements that could help you to construct your own models. Yeah, and one thing that, since I mentioned neural model and synaptic model, you have to bear in mind that, as in any other part of science, you can have very simple models, or you can have very detailed models. By detail, I mean biophysically detailed models. So the number of elements that you put in the model is large. So you can have simplified models and detailed models. Simple models, as in any simple model in physics, they are characterized by few variables. So a simple model, by definition, is something that has very few variables. So it's easy to understand them mathematically, to do calculations, analytical calculations for them, very few variables and parameters. On the other hand, a detailed model has many variables and parameters. So it captures, say, more detail about the reality, the real thing, but also it has the counterpart of having many variables and parameters. Those types of models would span all spatial scales that I mentioned at the beginning of the talk, from the microscopic from the microscopic to the macroscopic one. So that's why a model is also important. Biologists don't always understand this, that one does a model, one constructs a model, and the model incorporates elements from microscopic, say, realm to the mesoscopic realm and macroscopic realm, and then incorporates all of them, interrelate all of them, and it allows us to understand the effects from one scale to another one in a way that's very difficult to be done in other ways. So that's one of the reasons why models are important in biology, and biologists not always understand that, because they always want to work on the real system. Yeah, that's all. That's all for the first lecture of today. I think, yeah, finished in time. So that's it. So questions? One has 1 minute for questions, or we can continue tomorrow morning. I understand there'll be some time for us to talk. But anyway, you still have 1 minute to question, and this is just no. Yeah.
So what would be the difference